All right, well, thank you very much to the organisers for today. My name is Kieran Campbell. I'm a final year PhD student at Oxford. I actually submitted my thesis a couple of weeks ago, so apparently this is what I do in my spare time. This is joint work with Chris Yao, who is my supervisor, who's since moved to the University of Birmingham. And today I'm going to be talking about this idea of pseudotime, which you might have heard a lot in the context of single-cell analysis and single-cell data. And we're thinking what happens when we apply these sort of models to population-level data sets, in particular from population-level cancer genomics data sets. So what's the motivation for this? Well, the idea is that when you look at disease studies, there's only two data types. You normally either have longitudinal data, where you follow patients over time, or you have cross-sectional data, where you have a snapshot through the population. But one of the difficulties is that longitudinal studies are expensive, they're hard to do, you have things like censoring, and lots more work. But if you have a cross-sectional data set, there's a sort of asynchronous nature to it, where even within that data, there are patients at different levels of progression, right? So some patients within this data set will have sort of early disease progression, and some patients will have late-stage progression. So the idea is, can we exploit this asynchronous nature of the data to try and relearn some of the temporal information there? And in the context of molecular omics data, so things like gene expression, this essentially allows us to learn a continuous trajectory through the data space. So the motivation for this, as I said, comes back to single-cell pseudotime. So in the last sort of 5 years, you can now take single cells and you can measure the gene expression in each cell. But quite often what happens is these cells are undergoing some temporal process. So this is often differentiation, where I start off with some sort of stem cells here, and they differentiate into some cell type at the end. But what happens when I have the capture process is I actually lose this temporal information. I lose any idea of where each of these cells are. And so the whole idea behind these pseudotime algorithms is, can I take my raw gene expression measurements and reconstruct this trajectory, where essentially I give each cell its own time, and each cell becomes a surrogate time point for what I think my gene expression profile looks like at that stage. And there's been several talks so far in various sessions at this conference examining this idea. So the basic way we approach it, there are many methods, there are sort of many motivated by ideas in computer science, but we approach it from a statistical latent variable perspective. And how this looks is I start with some gene expression measurement Y, so this is a high dimensional vector measured across normally the entire transcriptome. And I want to explain why in terms of this latent pseudotime, which stands for the progression or encodes the progression of the cells. And to do that, I sort of draw a pseudotime, if you like, and I pass it through some mapping function, and I think that's how I produce Y. But the difficulty here is all I'm given is Y. All I have is a gene expression measurement. So I need to somehow infer, or at least make assumptions about, both what the pseudotimes are and how this mapping function looks. So for example, if I choose a linear mapping function like here, what I'm essentially saying is, as my cells progress, or as we think about later on as my disease progresses, I expect my features, which is my gene expression, to change linearly over this time, right? I can also consider nonlinear variants. So for example, I could think of parametric nonlinear functions like sigmoids, but I could also think of non-parametric nonlinear functions like Gaussian processes, where essentially what I'm doing is defining a probability distribution over all the possible mappings. And these normally take the idea of some sort of smoothness criterion. So I'm expecting a function that's smooth to some extent. So we've looked at this before. This is published work here, where essentially we've looked at fitting these pseudotimes for single cells using Gaussian process latent variable models. So essentially what you get is, in this sort of reduced dimension representation of the cells, where each point here is a cell, I learn the trajectory passing through this. And what I learn essentially is a probabilistic curve that says what's the probability that each cell is at this stage through the trajectory. We've also looked in terms of branching. This is becoming a really popular way of analysis, where what happens if I start with the cell here, it sort of differentiates a bit and then makes a decision. It sort of makes a cell fate decision and bifurcates into two or more types. So you can imagine this sort of stem cell might come along and differentiate into different types of neurons or that sort of thing. And this is important if we scale this up to sort of population-level data sets, where I'm looking at disease trajectories, where I can imagine that such a bifurcation might correspond to patients who recover well or patients who relapse or similar things. And this is further published work here. So the whole idea is we have this motivation from single cells, where we have this sort of asynchronous nature of progression, and we learn this trajectory through the data. So what happens if we apply this to population-level studies? And essentially what I'd end up with is I'd be able to track patients along these using cross-sectional data, and I would be able to molecularly stage the diseases. And a similar concept, I can sort of stratify patients. I can begin to stratify patients based on whether they're early or late stage, and I can also think about things if there are these bifurcations in the data. And furthermore, you can imagine this being predictive of the outcome for an individual patient. This actually builds on previous work. So funnily enough, the original pseudotime algorithms were designed for bulk level cancer progression. And then we've had this sort of flourishing of the single cell field, and we're almost trying to bring it back a bit. But the question we consider here in this talk is that when I have a single cell case, all my cells progress sort of uniformly along this, right? They're sort of cultured cells, and I expect them all to be the same to some extent. But if I apply this to population level data, then well, we know, we hear all the time that each individual is different, right? Each individual sits in a different genetic background, perhaps there's different phenotypes, or different environments as well. So how can we come up with a model that deals with this in the trajectory case? And to do this, we've come up with a new type of statistical model, or at least we think it's new. We call it covariate adjusted latent variable model. And I'll take you through it quickly here, but we have a more sort of pictorial representation on the next slide. And essentially what happens is I have my gene expression vector Y again, but I also have an additional matrix of covariates, which stands for these sort of phenotypes or environments that I couldn't measure before. So this might be things like mutation status in a particular gene. It might be things like sex or age. So how I model it is I model gene expression in terms of just standard differential expression plus this sort of progression-dependent term that's dependent on the progression of each sample. But crucially, this variable C here is gene-dependent. It tells me how the gene evolves over the sample. But this evolution can be modified by the covariate. So that sounds a bit abstract, but if we go to the next slide, this might give you a better idea of what's going on. So I start with gene expression in a mixed sample where I might have red and blue phenotypes sitting here. So I model this in terms of differential expression. So here, blue is higher marginal expression and red is lower expression, and some sort of latent progression, right? So I can imagine the latent progression. I could have the case where the features don't evolve at all and they're sort of static with respect to this progression. But I can also envisage a case where the genes are differentially expressed along this latent trajectory, right? And in particular, this might be different depending on the covariate of interest. So for here example, the blue people or cells or whatever are upregulated along the trajectory, while red has no change in expression. And what this model allows me to do is it allows me to learn this latent trajectory in the presence of these confounding, not necessarily confounding, but sort of a non-homogeneous genetic background. And it also lets me pick out interactions between this gene expression and the trajectory for the different phenotypes of interest. And inference is performed using variational Bayes. It's very fast. It scales up to thousands of samples, and you can do the whole transcriptome pretty fast on your laptop computer. So just as a quick example, one of the things we applied this to is the breast cancer RNA-seq dataset from the Cancer Genome Atlas. What you find when you fit the model is that this pseudotime, this trajectory, actually corresponds to aneurogenic pathway activation, which is vascular growth, right? So you essentially identify molecular progression of samples, where at one end I have patients who have low expression of the vascular growth pathways, and at the other end I have high expression of vascular growth pathways. And for the sort of non-homogeneous genetic background, this red or blue here, we chose ER receptor status, which is whether the tumor has an ER receptor, which sort of modulates the disease outcome and is known to. So as I said, you find that this corresponds to vascular growth. It wouldn't be a bioinformatics talk without some Go analysis, so you can look at which genes vary along this. But to give you an idea of what the model actually picks out, if I look at this molecular progression, this vascular pathway score here, if I take the gene FBP1, what you find is that if I'm an ER-negative patient, this gene is upregulated as the vascular pathway turns on as you molecularly progress within this pathway. Whereas if I'm ER-positive, you find that this gene turns off. And you find the opposite example for the FOXC1 gene, for example. But because this is transcriptome-wide, I can do this across all the genes at once, and I pick out this sort of landscape of interactions where I find genes associated with vascular growth, but I also find this covariate pathway interaction sitting here, where I have genes modulating the association with the pathway. So as a positive control, you can check out some vascular growth factors. So you see things like FGF2 are upregulated as you sort of drive along the molecular progression of your samples here. One particularly surprising thing we found was that if you look across many genes, and I mean like thousands of genes here, you find that if you have this low pseudotime, so if you have sort of less molecular progression of your sample, you find that your ER status dominates your gene expression program. So for example, if you look at FOXA1 here, if I have low vascular pathway score, what I find is FOXA1 is highly expressed in ER-positive samples and lowly expressed in ER-negative samples. But as is this molecular progression in passes, this actually converges to this point where ER status doesn't actually matter anymore and only vascular pathway does. So the point here is this could have very real implications for treatment decisions where if I'm looking at this molecular progression, there's no point in looking at ER-based therapies towards the end point because there's no impact on gene expression programs there. So just to wrap up, it's possible to do some sort of time series modeling or some sort of molecular progression modeling without explicit time series data. We have this covariate modulated progression models, and it allows you to build in pre-existing biomarkers alongside genomics measurements. We have an implementation of this that runs on existing data sets. It's on Bioconductor currently, so please give it a go. This is currently up as a preprint as of a couple of weeks ago, so feel free to check it out if you're interested. And the software is called Phenopath. It's on GitHub. It's now also in Bioconductor. It works in the sort of Bioconductor verse, if you like. Just to say thanks to my supervisor, Chris Yao, and various people who funded this. And please get in touch if you'd like to find out any more. Thanks.
Thank you, Kieran. We have time for questions.
So I have a question. In your model, you assume the error to be Gaussian, right?
Yes.
And did you think about implementing other distributions, as gene expression might be negative binomial or certain other distributions?
Yeah, so we're going with the sort of assumption that the log gene expression is approximately normal, which is used in various software. The real reason we like it is it makes the model conjugate, so you can do this really fast inference. It works well. I wouldn't sort of use it as a alternative to differential expression tools, because obviously the raw counts are a negative binomial, right? But if you look at models like Limavum or Sleuth, they have this log normal approximation as well, and it appears to work okay.
Thank you.
Thank you, Kieran. So our